Cell injury. Cell injury. Cell is the basic structure and functional unit of the body. Cell injury is defined as effect of variety of stresses that cause changes in cell's internal, alteration in cell structure or functions as well as external environment. The cellular responses to stress depends on variety of factors, and also the cell injury can be either reversible or irreversible. When injury is severe, it may lead to cell death. Cells may respond in 3 ways to any stress. Various forms of cellular responses to cell injury. 1. Cellular adaptation. Cell may adapt to the changes expressed morphologically. Revert back to normal after the stress is removed. Reversible cell injury. 2. Reversible cell injury. Mild to moderate stress, injured cells may recover. 3. Irreversible cell injury. Persistent injury, cell death may occur. 4. Subcellular changes. Residual effect of reversible cell injury may persist in the cell. Cell injury at subcellular level. 5. Intracellular accumulation. Persistence of reversible cell injury, metabolites may accumulate in the cells. Etiology. Causes of cell injury. Cells may be broadly injured by 2 major ways. A. By genetic causes. Genetically inherited and teratogens. Developed defect starting at fetal life. Cytogenic defect. Abnormalities of chromosomes. Single gene defect, multifunctional inheritance disorder. B. By acquired causes. Hypoxia, deficiency of oxygen, and ischemia, reduced supply of blood to cells, anemia, co-poisoning, cardiorespiratory inefficiency, etc. Physical agents. Mechanical trauma, e.g. road accidents, thermal trauma, e.g. heat and cold, electricity, radiation, UV and ionizing, radiation, UV and ionizing, rapid changes in atmospheric pressure. Chemical agents and drugs. Chemical poisons such as cyanide, arsenic and mercury, strong acids and alkalis, environmental pollutants, insecticides and pesticides, oxygen at high concentration, hypertonic glucose and salt, social agents such as alcohol and narcotic drugs, therapeutic administration of drugs. Microbial agents. Bacteria, fungi, protozoa, virus, other parasites. Immunologic agents. Hypersensitive reactions, anaphylactic reactions, autoimmune diseases, immunologic diseases. Nutritional derangements, malnutrition, deficiency, and nutritional excess, obesity, atherosclerosis, heart diseases and hypertension. Psychological factors. Mental stress, strain, anxiety, overwork, frustration, alcoholism, smoking. Others. Aging, idiopathic diseases, etc. Pathogenesis of cell injury. Injury to cells may have many causes. In such case, oxygen plays a central role in cell injury. Mechanisms of cell injury includes hypoxia and ischemia. Cell injury. Cell membrane damage, mitochondrial damage, ribosome damage, nuclear damage. Cell membrane damage. Physical agents such as heat or radiation can damage a cell by literally cooking or coagulating their contents. Impaired nutrient supply such as lack of oxygen or glucose or impaired production of adenosine triphosphate, ATP, may deprive the cell of essential materials needed to survive. Mitochondrial damage. Increased in cytosolic calcium, coupled by an increase in inorganic phosphate and certain fatty acids. High inorganic phosphate and fatty acids alone cannot damage the mitochondria, but coupled with high calcium are extremely damaging to cell. High calcium alone can still damage mitochondria. Ribosome damage, leading to altered protein synthesis. If hypoxia continues, intracellular protein synthesis decreased due to damage to ribosomes and polysomes. Continuous hypoxia causes cytoskeleton changes with loss of microvilli and formation of blebs on surface of cell. This swelling results in swelling at mitochondria and endoplasmic reticulum. Nuclear damage. Decrease in cellular ATP and increased in AMP also stimulates the enzyme phosphofructokinase glycolysis in order to maintain the cell's energy sources by generating ATP from glycogen. This stage is reversible if oxygen is restored. Pathogenesis of reversible cell injury, hypoxia, ischemia leads to decreased generation of cellular ATP. Sodium-potassium pump damage leads to accumulation of sodium and water inside the cell, which ultimately cause cell swelling. Increased anaerobic glycolysis leads to increased formation of lactic acid, which cause clumping of chromatin. Detachment of ribosomes from endoplasmic reticulum leads to decreased formation of proteins. Detachment of ribosomes from endoplasmic reticulum leads to decreased formation of protein. Pathogenesis of irreversible cell injury, permanent cell injury. increased calcium influx, mitochondrial damage. Phospholipase activation, membrane damage. Protease activation, protein damage. Endonuclease activation, nuclear damage. ATPase activation, ATP damage. Bursting of lysosomal enzymes, cell death. Damage to lysosomal membrane, liberation of hydrolytic enzyme, digestion of cellular components, nuclear, damaged or changes, leads to cell death or three states. Autolysis, self-digestion, disintegration of cell by its own hydrolytic enzyme. Necrosis, focal death of living tissue, progressive degeneration by various enzyme. Apoptosis, coordinated, internally programmed cell death.